Okay, hi everyone. So today we'll learn how to use MELD to predict protein peptide binding. So MELD is a Bayesian framework-based plugin to OpenMM molecular dynamics software. MELD helps us to integrate data from different sources like experiment, bioinformatics into computer simulation in order to study binding or folding of different biomolecular systems. So in this particular example, we'll learn how to use MELD to predict native structure of protein peptide binding. So for that, first thing we need to know how to install MELD. So this is a GitHub page for MELD, and here you can see the installation instruction. So you will need to install Miniconda first and then follow this couple of steps. And it's pretty robust and fast. And there is another video showing how to install it. That should be available here. So once you have installed MELD, now let's go to the tutorial. So the link will be shared with the video. And so protein peptide tutorial, and the README file is here. Okay. So for this particular example, we have used a protein peptide complex which has a known structure, experimental structure. So the protein here is BRD3 protein from ET domain, which binds with MELD peptide. So this MELD peptide is unstructured when unbound, and when it binds, it gets structured. Well-known 3D native structure with beta strand. So these are the PDB code given here, and the MELD version used for this work is, this is important to know, this is the MELD version 4.2.0.4.20. And okay, so about the system. So this is the protein sequence we had, which is 96 residue long, and 28 residue are like purification tags. So we will remove that. We'll have 68 residue. And peptide is, I guess, 18 or 17 residue long here. So the protein, as I mentioned, peptide is unstructured when unbound, and when bind, it gets structure. And the protein also experience some conformational change upon binding. So this is example of folding upon binding. So basically, first of all, how to set up the simulation. So once MELD is installed, you can use this piece of code to set up a PDB file from the peptide sequence. If you have to write this sequence file of the peptide sequence.dat and run this code. So I have another tab open here showing the files needed for this. So the piece of code you will need is this. So this will sequence.dat, so you will need to run it. It will use first build the PDB and use Amber to minimize the PDB structure of peptide. So after that, you will have minimized protein as well, combine them together in a PDB file, and make sure the peptide is at least 30 angstrom away from protein. So we don't want to be in interaction site before starting the simulation. So our starting point looks like this. Here is the protein, and the peptide is far away from the protein. To change the coordinate of the peptide, I have used the code here listed, but there are plenty of different ways to do it. Okay, so now come to the MELD simulation. So as I mentioned, MELD help us to integrate data into the simulation. So in real world, we will have some experimental data or some bioinformatics data. But in this case, what we're using, since it is just to learn, we are using native structure. So we know the native contacts. So basically we'll back calculate the native contacts and use them to guide the simulation. So we'll need two kind of restraint like input file. One to, because protein is already known protein structure, so we don't want to change the protein structure a lot. So we'll fairly keep the core of the protein structure fixed. So that's why we will have intra-protein residue contact. So C alpha, and we'll use binding restraint, which is protein and peptide residue, which are within 8 angstrom. We used mLiters to calculate this restraint. The restraint file look like this. Let me show you here. So this is for the protein only. So this means first residue C alpha, fifth residue C alpha is 6.6 angstrom away, and so on and so forth. And notice that we have a space after each restraint, and that is important here. I'll explain later in the code part. And this is for the protein peptide binding restraint. So this means fifth residue C alpha and C alpha on 82nd residue are 0.58 angstrom away. So basically this is calculated using mLiters, but it can be any experimental data again. And these two are the distant file. So we'll use them. So once they are ready, we have the code here as well, how to generate them. This is to generate the protein contact, and this is to generate the protein peptide contact. So now, once we have all this, we can go to set up a MELD simulation. So we need two of the input restraint, one minimized starting file, and another file we need to set up MELD is that setup MELD.py file, which is essentially configure the simulation. So in that file, we import all the modules we need. Then this is the main file which set up the replica ladder, assign it temperature, force constant, read the input files, launch OpenMM job, and does almost everything. And here we assigned number of replicas, how many replicas we need here, in this case 30. Number of steps of the simulation 2000, which is around 100 nanosecond. Notice it's very little, but since we are using native contact, it should be enough. Here are some functions to define the system. This is first initiate the system. This function is to read the input file for binding. So each of the line you noticed here, you saw here, basically one of this each of the line is separated by space. So whatever we have separated by space is called a group, and inside a group we have restraint. So basically here with this group we have just one restraint. So we'll trust certain percent of this group in case of experimental data. But again, since this is native contact, we know all are correct. So basically we are reading them through this function, and we are trusting one group in one restraint because we have only one here, and that should be true. And here that's how the energy penalty works here. From zero distance angstrom to whatever angstrom we have, there is no energy penalty. And then after that, we have up until 2 angstrom. After that distance, we have energy quadratically increasing. And then after linear, and with the force constant 350 kilojoule per mole per Newton square, nanometer square. And here, this is another function to read the protein contacts. Here, since we want to fairly keep the protein to fix to its initial structure, we enforce this restraint more strictly. Here, if you notice here, the flat bottom, that means the energy penalty is 0, only if this enforce the distance from MELD simulation is 2 angstrom, with 1 angstrom less or 1 angstrom more. Beyond that, it's harmonically increasing. Here, how to set up this, how to call these functions. Here, it's reading the template file, starting file from template directory, and using this force field, FF14SB side, which is combination of 2 force field, this backbone from 14SB and sidechain from 99SB. Here we are defining the replica ladder, temperature replica ladder. So these values goes from 0 to 1. 0 means first replica, 1 means 30th replica. In this case, we are using 0 to 0.4. That means 0 to 12 replica. Temperature goes from 300 to 500. And there are some scalar we are defining here, which will be used later. And here we are reading the binding restraint. Notice this is non-linear scalar. That means from replica 0, first replica to 12th replica, the force constant is the maximum. And then 13 to 30 replica, it is decreasing slowly, eventually making it 0 at 30th replica. And we are trusting all the contacts basically with this is 100% of it. And this is for protein contact, where we are using constant scalar. That means the restraints are always on throughout the 30 replicas, and we are trusting 90% of it, because we want to give little flexibility to the protein, because there might be some change in the conformation. Now these are pretty standard. We don't change this generally. This is the solvent model we are using. GBNEC2 implicit solvent model. This is the how frequently we save the frame, which is 50 picosecond. And these are like how fast we change, how frequent we change the replica exchange, and all those things. So these are not always need to be changed. We can keep them fixed as it is. And these are well optimized values. Okay, so so here I put the code as well. You can look at here. So you might, instead of inter-residue contact in the protein to fix the protein fixed to its initial conformation, you might want to use different kind of restraint, which is called Cartesian restraint. You might want to restrain the coordinate of the C alpha or any atom in the protein. In this case, this is the function we will use, and this is how we call this function. We have to define the range. This is the range of the protein residue. We want to fix the protein, and then we'll use constant scalar again, because we don't want the protein to unfold. And this is always active. That means in every, we are trusting all of the C alpha atom here in every frame. So once we have done it, we have to execute this code. And let me show you in the terminal. Here I have this. I have this file, this protein peptide file, this protein contact file, this setup file, this template. Template has this minimized complex starting point. And these two essentially will get later. Since I already have result, I have this initially. You will not have this. I have already load MELD module. So let's do this. So you will see some harmless complaint these are. So this is setting up the replicas. It has 30 replicas, so you will see 29. So if you do this, you will see it's created data directory, and it has this. So once you submit the job in queuing system, it will create trajectory.pdb here. So to submit the job, go back here. You will have this. This is the command which does it. I think I have the job script, how to submit it. or I guess I don't have it because uh, it changes depending on the queuing system you are using. So there should be some headers of whatever queuing system you're using. So notice that this will always need multiple GPU because you are using replica exchange. So you will need at least 2 GPUs in order to run this job. So here, if the job fails, you can run this command and do this again. It will restart the job. So after that, it will create remd.log file. You can print the end of that file to see the real-time progress of this job. This file will be generated. And when the job is finished, you want to do several things because this MELD trajectory produced by this simulation will have everything encrypted inside, like 30 temperature replicas and 30 walkers. So we can extract them. So to extract temperature replicas, we have to execute this command. This 0 meant it will extract the first replica. And if you execute this command, it will extract the first walker. So the best thing to do this to submit this. These are not very short. This will take time. So it's better to submit them in the queuing system. So I have already extracted the first lowest temperature replica. Let me show you how it looks. So this topology file I have saved from tleaf while I was setting up the system or minimizing the system, initial system. So you also need this topology file to read this file. So the protein is 68 residue. So start from the beginning. So initially it was like this. The peptide is far away. This is now, as you go, it's hard to control this VMD in supercomputer. So it, as you notice, it binds very, like within very few steps. That's because we are using the native contact here. In real case, you might need to run it more than 1 nanosecond, sorry, 1 microsecond or 1.5 microsecond. So here is the RMSD of the trajectory, first trajectory. You can see within just 200 frames, it comes really close to the native. And here, then what we do clustering, hierarchical clustering on 5 lowest energy trajectory, lowest temperature trajectories. And this is the native, and this is our prediction. And this prediction is essentially the centroid of most populated cluster, C0 of C. So C0 is our prediction. So basically, so now if we overlap them, you can see they're matching pretty well here. And there are several other analysis that you can perform to see whether this simulation is optimal. The protocol is optimal, whether the replica exchange are good. You can execute this command first. It will show how both of the command will show how good replica exchange is. First one will show something like this. So this is an example of good replica exchange. This is the poor replica exchange. You can see the colors are mixing well here not. Different color represent different replicas. And here, if it's from second command, you will get this. So the closer to the dashed line is better. And you can see this defined poor replica exchange. So there can be many other analysis done, but that depends what kind of goal we want to achieve. But for this particular example, our goal was to predict the native structure, and we have predicted here successfully. And with that, it will end here. Thanks everyone for listening.